So welcome back, everyone. So we will have now our next talk by Nina Luning Prak, who is a professor at the pathology and laboratory medicine in the Perelman School of Medicine at the University of Pennsylvania. And Nina is going to present her work on B-cell clonal networks and the lessons learned from SARS-CoV-2.
Hello, everyone, and welcome to my talk. I want to start by thanking the organizers, especially Encarnita, for putting this session together. My name is Nina Luning Prak, and it is my privilege to give you an update on some recent work that we have been doing to look at B-cell clonal networks and some of the lessons that we've learned from SARS-CoV-2. These are my disclosures. In addition to my consulting work, I also direct the Human Immunology Core at the University of Pennsylvania, where we perform immune repertoire profiling on a fee-for-service basis. In today's talk, I'm going to cover three different small vignettes. The first is to describe for you features of expanded clones in hospitalized patients during acute SARS-CoV-2 infection. In the second part, I will describe for you some expanded clones that we have been studying in individuals who have recovered from SARS-CoV-2 and were subsequently vaccinated. And then in the third part, which is brand new and completely unpublished, I will describe for you our initial analyses of tissue distribution of expanded clones in a COVID autopsy case. So in the first part, let's dive right in and talk about features of expanded B-cell clones in hospitalized patients with acute COVID. This work builds on two previous studies that we performed on hospitalized COVID-2 patients at the University of Pennsylvania, and the references are shown here. So in this analysis, we studied whole blood from hospitalized SARS-CoV-2 patients. We looked at three groups of individuals: healthy controls who were not hospitalized, and then COVID patients who either had moderate COVID, meaning they were hospitalized but not in the ICU on a ventilator, or severe patients who were in the ICU. We studied the B-cell repertoire using antibody heavy chain gene rearrangement analysis from whole blood. And here what we're looking at are the top copy clones and asking how large are they in COVID patients compared to controls. So in the first graph on the left here, you can see that the top 20 most frequent clones occupy a much larger percentage of sequence copies in the libraries of COVID patients than healthy controls. Here, each dot is the percentage of copies found in the top 20 ranked clones for each patient. If one takes this D20 fraction and plots it as a function of the plasmablast percentage, you can see a very nice correlation. And so we infer from that that the plasmablasts are highly enriched for these expanded clones. But this was at a time when we were not allowed to sort yet for biosafety reasons, so we could not directly prove this other than by correlation, which of course is indirect. Next, we looked at the top copy clones and analyzed their level of somatic hypermutation. So here each dot is a clone, and we're looking at the level of mutation by its level of identity with the nearest germline V gene sequence. So 1 means a germline or unmutated sequence, and lower fractions mean more mutation. So COVID patients have higher levels of mutation in some of their clones, but they also have a sizable fraction of clones that are unmutated. Similar findings have been reported by other groups, including notably the work from Scott Boyd's lab. In addition to there being large clones, the other thing we noticed was that these large clones tended to have long and variable CDR3 sequences. So shown in the plot on the left here are the CDR3 lengths of the top 10 clones in each person. And so again, each dot here is a clone. And you can see that in the severe COVID patients especially, there are several clones that have much longer CDR3 lengths. And if you look at the sequence composition of these large clones at the amino acid level within the CDR3, what you see is quite a bit of diversity in terms of the CDR3 sequences. And not only are these CDR3 sequences diverse and long, but they also tend to mostly be private. So in this plot on the right here, we're looking at private versus public clones. And the public clones tend to have shorter CDR3 lengths. Shorter CDR3 lengths are also more likely to be shared. And the private clones tend to be the ones with the longer CDR3 lengths. So in this first part, I wanted to share with you some general properties of large expanded clones in hospitalized COVID patients. These clones are large. They seem to be correlated with the plasmablast fraction. They have long CDR3s. They tend to have mostly private CDR3 sequences, and they have variable levels of somatic hypermutation, and many of them are unmutated. And the low level of somatic hypermutation may reflect an extrafollicular immune response, or one which may not require T-cell help. And this is consistent with some of the other studies that have also suggested that there is an extrafollicular immune response early in COVID infection, and particularly in severe COVID. And that this extrafollicular response could be similar to a disease flare that you might see in autoimmunity, such as lupus. And so along those same lines, hospitalized patients with COVID do also produce autoantibodies. And in our work shown here in this panel on the right, but also in a recently published Nature paper from others, there are many different autoantibodies that hospitalized patients with COVID produce. So perhaps these two findings are related, although we need to do more work on this to actually directly prove that. So turning now to the second part of my talk, I'd like to describe for you some analyses that we have performed on expanded B-cell clones in individuals who have recovered from COVID-19 infection and subsequently received an mRNA vaccine. So this work also was recently published and describes distinct antibody memory B-cell responses in patients who are either naive to SARS-CoV-2 or who were previously infected with SARS-CoV-2. So the basic outline of this study is that we're comparing individuals who are naive, shown in blue, to individuals who previously had SARS-CoV-2 infection, and then sampling their blood over a period of time relative to the vaccine doses. So in time point 1 is at baseline prior to the first dose of vaccine, time point 2, 14 days after the first dose, time point 3 is at right before the second dose, and then time point 4 is 7 days after that. And we're looking both by serology as well as by cell-based assays to evaluate the immune response to the vaccine and compare it between these two groups. So if we start off first by looking at the serologic findings in this study and compare individuals who have recovered from COVID infection, shown in red, and here each line indicates a person, to individuals who are naive to COVID and are receiving the vaccine. It's very clear that people who have recovered from COVID already have evidence of spike-binding IgG antibodies at baseline prior to receiving the first dose. Not the case for naive individuals, as one would expect. But then what's really interesting is that once the recovered individuals have received that first dose, they bump up their antibody levels, but then they stay put. And the boost dose, so at time point 3 and also time point 4, you do not see an increase in antibody levels. In contrast, the naive individuals have an increase in antibody levels after both doses of vaccine. Turning to the B-cell response to the spike protein, if one looks by flow cytometry at the proportion of memory B cells that bind to the spike protein, again with red dots signifying patients who were previously exposed to COVID, blue being naive individuals, again, the same pattern is observed where the boost does not increase levels of spike-binding B cells in the recovered individuals, but makes a large difference in the naive individuals, where you see an increase in spike-binding B cell fraction both after the first dose as well as after the second dose. So we wondered at the B cell repertoire level what accounts for an increase in the fraction of spike-binding B cells in recovered individuals. And there are two general explanations. One is that there could be recruitment of new clones into the response, or the second possibility is that you have expansion of pre-existing spike-binding clones. And of course, these two possibilities are not mutually exclusive. So as a starting point, we looked at the clone count, the number of spike-binding clones that we were able to recover over time in 5 individuals who were previously exposed to COVID and received an mRNA vaccine. Here, each line is a person, and you can see that there's quite a bit of heterogeneity in the clone counts. In the most sampled person, P20, you can see that there are clones that are found at single time points, but also clones that overlap between the time points. So we thought, okay, we're not going to get too far with clonal overlap analysis other than to say that we have a lot of different clones that people are making, and in some cases, people may indeed be recruiting additional clones into their response. So what about the clones that were there from the beginning? Do spike-binding clones that were there from the beginning evolve following vaccination in recovered individuals? So to look at this, we focused on clones that overlap between time point 1 and at least one other time point. So if clones evolve following mRNA vaccination, we would have expected to find larger numbers of somatic hypermutations in clones at the later time points in recovered individuals. So in the graph on the left, we're looking at the level of mutation in overlapping clones that bind to the spike protein, and we can see that there's no clear increase in somatic hypermutation with time. On the right, we're drilling down on individual clonal lineages and asking whether we see more evidence of higher levels of mutation, which would be shown by having nodes within the trees that are more towards the leaves of the lineages, so further down in the lineage. For example, here's one where, at time point 4, we have a node that has 12 mutations in the V gene sequence compared to the nearest germline gene, and another node here at time point 2 that has 17 mutations away. So no convincing evidence for more mutation at the later time points. Here's the lineage where you see no change at all in terms of the members of the clone. All of them have similar levels of mutation in the V gene, and they're all in one node of the lineage. So in terms of the just brief conclusions for part 2, recovered individuals increase their antibody titers, also in data I have not shown you, but are present in the paper, their virus neutralization titers, as well as their proportion of circulating spike-binding B cells following dose 1, but not the boost or dose 2 of the vaccine. Therefore, it may be sufficient to give people who've been exposed to COVID a single vaccination. And this, of course, was a very timely topic when vaccine doses were limiting. However, in the future, it may also suffice to give a single booster to vaccinated individuals, although this needs to be studied further. Since we have not looked at that here directly, we've only looked at recovered individuals, not previously vaccinated individuals. One of the major things that we're very interested in this analysis is to try to understand more about the mode of memory in people. Is it really clonal recruitment rather than somatic hypermutation, or is there also more somatic hypermutation if we look later on? Which is in fact what we see in COVID recovered individuals. You can see more somatic hypermutation occurring over a period of months, not days. And so this is something that we are certainly interested in pursuing in future studies. We're also, of course, very interested in understanding more about the effects of virus mutants on these responses. I want to mention that for parts 1 and 2, these data sets are available in SRA and on the Air Data Commons, and you can see here the PMID number as well as the SRA project number. And for more information on the Air Data Commons, here's a website for you to visit. So in the third part, I want to focus on tissue distribution of expanded clones in an autopsy case of COVID-19. And so this particular case that I'm going to focus on today is an elderly male who has a past medical history that is significant for prior history of deep venous thrombosis, pulmonary embolism, who also had hypertension and gout. His history of COVID consisted of weight loss and cough and fatigue for over 3 weeks prior to admission. At the time of admission, he rapidly deteriorated, and several of his problems included having acute kidney injury, cytokine storm, amyloidosis with focal mixed myocardial infiltrates, septic shock, and multi-system organ failure. Just some histology here, care of our surgical pathologist colleagues, Kathy Montone, And you can see, for example, in the lung, there's a obviously not normal looking diffuse alveolar damage with, and if you look under higher power, you can see a mixture of different cell types, including plasma cells, immunoblasts, and histiocytes. Similarly, the kidney is also not looking very good with probable acute tubular necrosis, but there's also evidence of chronic kidney disease, both by history and by histology, with interstitial fibrosis, glomerulosclerosis. There's patchy chronic inflammation in the interstitium, and it's not really clear how many of these findings are specifically related to COVID per se, as one can see some of these findings in other autopsy scenarios as well. So just a brief overview of the V gene usage. You can see VH169 sequences in the lung are quite prominent, and so we immediately kind of knew to focus there. So over 20% of the sequences in the lung from this patient were VH169. So we started by looking in the lung. And so here again, we're doing bulk gDNA-based sequencing of antibody heavy chain gene rearrangements and looking at the top 20 most abundant clones in the lung. And so what this plot shows you is that there are some large clones in the lung. For example, this top-ranked clone here comprises over 5% of the sequence copies of the libraries, sequencing libraries in the lung. And the top 20 clones collectively occupy over 40% of the sequence copies of the lung antibody repertoire. So if we now look at that top copy clone and ask where is this clone distributed in the body, it's a very interesting result. What we see here in the clonal lineage is, first of all, that the clone has somatic mutations. So the trunk of this lineage is 14 mutations away from the nearest germline V gene. And then the nodes here of the lineage are colored by tissue. And the size of the nodes gives you the copy number fractions. So you can see here that a lot of these early nodes are in pink, which corresponds to lung. And then the node at the bottom is mostly in the kidney. And recall that this individual had evidence of both chronic kidney disease, but also acute kidney injury. If we look at the V gene sequences sequences that are sitting at the trunk of this clone, shown on the top line here, and sequences that are found in the kidney predominant node, shown on the bottom, there are amino acid differences within the V gene. Furthermore, when we search through our entire data set on this individual, we find actually several additional clones that have very similar sequence features. You can see here highlighted in red are shared amino acids in the CDR3 sequence, and you can see that all of the clones have either VH1-69 or highly similar VH1-8, and then there's one that's VH1-46. So these may all be parts of a larger clone, or they may be different clones that are nevertheless highly similar to one another. So the answer is yes, there are additional high copy clones that have sequences that can be found in the kidney and or the lung. So again, the kidney sequences are in purple and the lung sequences are in pink. So we also wondered whether we would see a similar pattern of clones that overlap between kidney and lung among the T cells. So using the adaptive platform, we sequenced T cell receptor V beta gene rearrangements from these same samples from the same individual and analyzed their tissue distribution. Okay, so here is a stacked bar graph of the top 25 ranked clones, T cell clones, in the body of this patient. And you can see that the clones that are present in the lung, shown by these pink segments here, are widely disseminated. So you can find them in lots of other tissue sites. In contrast, some of the other high copy clones are found at single sites. So we're still in early days with this, but we're pretty excited, and we have evidence for large B cell and T cell clones that are present at multiple anatomic sites, but also that the B cell and T cell clones, some of them are shared between lung and kidney, where there is evidence for pathology at both locations. Could these clones be evolving and could, in the case of the B-cell clone, the accumulation of somatic hypermutations be contributing to an alteration in specificity that might result in an off-target effect on the kidney. So we're obviously extending this analysis to additional autopsy cases and using these clonal networks and antibody cloning and specificity to gain further insights into potential mechanisms of COVID-mediated tissue injury and also possibly autoimmunity. Finally, I want to thank the researchers, patients, and families involved in these studies. It was a tremendous team effort. Thank you very much. Hello.
Thank you very much, Nina, for this great presentation and this impressive work in such a situation. So I have a question, as you may expect, on the last part, on the T-cell part. So, so the clones that you saw, so you could find clones in the different tissues with relative different frequencies. Have you checked for any specificity or linked with other diseases, maybe autoimmune diseases, because there seems to be located in particular tissues?
Yeah, it's a great question, and certainly we're very interested in doing that. The direct linkage to specificity, unfortunately, we can't do with these data because these were done on tissue sections. So these are just bulk sequencing libraries, but certainly from an informatics standpoint, we're eager to mine public databases and look for some of the specificities of these clones that are found in different tissues. And again, right now, this is still very early. These are early days. We're interested in knowing whether these clones that are crossing between sort of two injured tissues in some way relate to the pattern of tissue injury. And so we're doing this on, of course, other COVID autopsy cases as well. So.
And do you have any idea? So of course you haven't checked this, but of the cross specificity between the TCRs clones and the BCR clones that you've been finding?
Yeah. So this is so our first thought was that we would do this kind of by localization, co-localization. So if we see that there's certain T cell receptors and B cell receptors that are kind of coming together in space, that would be really exciting. And so that's why some of those large T cell clones that have sharing between lung and many of the other tissues, we kind of look at that and go, I wonder, are they related in some way? But so far, we don't have an obvious way of knowing. So far, the T cell clones that we do see that are in the lung tend to be widely disseminated in, across a lot of the other tissues. So that could, of course, also relate to their, potential biology. They may be effector T cells that are kind of trolling around, in the body. They may be TEMRAs or something like that. But right now we don't know that. We are working on trying to link some of these data to single cell data, where we hope we will have much more depth, not only at the clonotypic level, but also, transcriptionally or potentially learning more about the subsets involved. So just like Lindsay, I'm eager to give you an update in about a year from now. I think we'll know a lot more.
Can I ask one more question? Do you have one? Yeah. So I have a question regarding the clonal distribution that you see, and especially the 169 that are high. Isn't it also true that in the healthy repertoire, 169 is quite high? So how significant is this for COVID? And did you compare it, with the control, for example, generation probabilities? Like what's the normal distribution of the VGen and how does it compare in this COVID patient?
Yeah, it's really interesting. So we do have a fair bit of data from organ donors who do not have COVID. And in organ donors, what you generally see is that in the blood, a lot of the VH1 family members are more frequent, whereas in the tissues, you can see certain certain Vs tend to associate with different tissues a little bit, at the family level, not so much at the individual V gene level. And so here you know, but each person can be quite different in that. And so I'm not trying to make a claim that VH1-69 is in some way special with respect to COVID per se. I think within this individual, VH1-69 is highly represented in the lung and was kind of an early tip-off that, you know, something is going on there. But yeah, again, in terms of frequency, yes, the VH1-69 certainly is very frequent. It's one of what we call the usual suspects of V genes that we often see.
Great, thanks. Super talk, very nice.
Thank you.
Yes, very nice. So we don't have additional question in the chat, so we can move